the air out of your lungs. All right, now it is very likely that most of you are familiar with the term cystic fibrosis, or you may even know someone that has been affected by this disease. In the United States alone, there are over 30,000 people living with cystic fibrosis, and around 1,000 new cases are diagnosed annually. Cystic fibrosis causes a variety of complications in the human body, but its signature symptom is the thick buildup of mucus in the lung, the pancreas, and many bodily glands. Now, in the lung in particular, this thick buildup of mucus, it's going to lead to a lot of respiratory issues and ultimately shortens the lifespan of all patients with cystic fibrosis. The current average lifespan for a cystic fibrosis patient is 37 years, and in our opinion, this is far too short, which has led us to propose a novel treatment and hopeful cure for cystic fibrosis altogether. However, with this being said, before you can diagnose and treat a disease such as cystic fibrosis, it is important to understand the variety and complexity of the CFTR gene mutations which directly cause cystic fibrosis. This is something that Gabby will be expanding upon.
So cystic fibrosis is an autosomal recessive disease, meaning that both parents must be carried in order for the disease to be expressed in an individual. There are 1,700 known mutations that cause cystic fibrosis. In all of these mutations, a mutated CFTR gene produces a damaged or defective CFTR protein. The CFTR protein has a vital function in many visceral organs of the body. It regulates the balance of salt and water in the surface of the body organs such as the pancreas, liver, and most importantly in individuals with CF, the lungs. It does this by acting as a chloride channel. It transports chloride ions across the membrane of epithelial cells that produce mucus, sweat glands, and digestive enzymes, meaning it's important in the reproductive, respiratory, and digestive system. It's also responsible for the mucus lining of these organs in these systems. So, therefore, if in individuals with cystic fibrosis where they have a defective CFTR protein, the mucus is thick and sticky, which causes difficulty breathing and also causes other lung-related illnesses such as pneumonia. This is why we chose to have the lung as the main focus of our grant proposal. And now Felipe will talk about how these 1,700 different mutations are grouped into different classes.
Okay, so these over 1,700 mutations can be classified into five separate mutation groups based on how they affect the resulting CFTR protein. Protein production mutations cause a protein that is faulty and just doesn't work. Protein processing mutations result in a protein that is in an incorrect 3D shape, which is an essential part of its ability to carry out its function. Gating mutations affect the CFTR protein to where it cannot properly open and close and allow the flow of salt and water in and out of the cell. Conduction mutations affect the efficacy of this flow, and finally, insufficient protein mutations lead to an inability of the cell to properly maintain the level of CFTR protein in the cell. For years now, pigs have been the chosen animal used to study cystic fibrosis symptoms due to their many anatomical similarities with humans. However, past studies have shown that pigs and humans have one key difference in showing CF symptoms, and that is in the lungs. Pigs do not show the same level of severe lung complications in the lungs as humans do, and being the number one cause of death in humans with cystic fibrosis, this obviously is something that we want to look into. Now, we want to use bioprinted human lungs in order to test the efficacy of new and novel treatments that are being developed currently to combat cystic fibrosis. And while you may think, wow, this is crazy, right? Having just an artificial lung outside of a body, that has to be super far into the future, right? Well, no. You might be surprised to know that in 2016, a team of Czech researchers was not only successful in creating this model, but they were able to keep it viable for an extended period of time. Now that we know that we can use this tool and it can be made, we can use it to not only test better treatments, but one day come up with a cure for cystic fibrosis. And now Ryan will introduce you to our objectives.
So we propose to create a cost-effective experimental design for personalized treatment for cystic fibrosis. As you can see in this table, a bioprinter costs significantly less than clinical trials, not to mention the kind of liability issues that come with clinical trials. So to do this, we will manufacture 7 fully functional bioprinted lungs, one with each of the most common mutated CFTR genes. Then we will test the effectiveness of different drugs on the diseased bioprinted lung and find out if it works for individuals with cystic fibrosis. Now Rio will go into more depth about our methods.
So our methods involve using a bioprinter by Frailus Biologics, which is the fastest printer on the market.
It has an unprecedented speed of about 105 capillaries in 6 minutes, which shows that time is not necessarily a constraint on our experiment. We hope to do so by basically creating a stem cell preparation by suspending stem cells in a sodium alginate solution, which basically allows us to create a 3D model of the lung in a similar manner as one would use Legos to build a house. It allows us to do it layer by layer, which allows us to construct a 3D model on which we can actually test different drugs on. We'll do this by creating 7 different models of the mutated lung, and we'll do this using the 7 most common mutations for cystic fibrosis. For example, the F508 deletion, which basically causes the ion channels to not open properly. So then we can create these several different models, and we can create different trials of them to basically test different drugs. So we can test like drug A on lung 1, lung 2, lung 3, and so on and so forth. And we'll do this using gene editing techniques, CRISPR-Cas9, which basically allows us to target a specific part of the genome and cut at the appropriate place to simulate a certain type of mutation. Then these stem cells will multiply and basically allow us to create an amalgamation of cells that are all mutated, which would simulate an actual mutated lung. We can then test different drugs and create a personalized medicine approach to this entire problem, which allows us to figure out which drug for which specific mutation is the most effective, so that in the future we can basically prescribe drugs to patients with certain mutations.
So by utilizing these 7 bioprinted lung models, each containing one of the 7 most common CFTR gene mutations, not only are we able to bypass clinical trials, we can start to create a personalized medical treatment for individuals based on their CFTR gene mutation, rather than simply looking at cystic fibrosis as one uniform disease. So by sectioning off cystic fibrosis into its 7 most common CFTR gene mutations, we hope to successfully prescribe the correct and most effective drug for each gene mutation, rather than prescribing one uniform drug that will simply ease the symptoms of cystic fibrosis in all patients. At a larger scale, we expect that the successes of our research will prove to be very valuable in the medical field as we continue to enhance our knowledge on bioprinting techniques and how we can bioprint human organs in order to learn how to better treat and cure genetic diseases.
So as with any kind of research or experimentation, there are potential problems that can arise. One, of course, is time. And not necessarily the time it'll take to bioprint, because as Rita talked about, Post Biologics has created an extremely fast bioprinter. But I'm talking about the time it will take to test all of our drugs. But this won't take any longer than clinical trials. A second problem is that the drugs don't work, which is definitely something that can happen. But this can actually be a positive, because first of all, we'll be making progress, and second, by learning that a drug doesn't work, we'd save someone from going through clinical trials for a drug that doesn't work. In other words, we can save someone time, money, possible negative side effects, and even save someone's life.
We proposed this research because, as previously stated by Felipe, this has only been done in pigs before. So nobody knows the real effects that it has on humans, the severity that it can have, and how to treat it. There's also no real cure for cystic fibrosis as of now, only treatments that lessen the severity of the symptoms people with cystic fibrosis experience. In the future, we would use these effective drugs, obviously, to treat patients with these genetic mutations, and as stated, we could target specific genetic mutations with specific drugs that we find through our research. This research will also advance research in the future as far as using bioprinting for other organs in the body and testing other diseases that affect those organs.
So in conclusion, what we're planning to do is create 7 different models of bioprinted lungs, each with one of the 7 most common CFTR mutations. By doing so, we can create an approach to treating cystic fibrosis that is personalized for the individual patient. This is a more cost-effective approach than taking these drugs directly to clinical trial. And by doing so, hopefully we can find a cure for CF.
Wow, cool!